Our last speaker, not of the session, but of the day, is Dr. Lindsay Gabbard. She is a microbiologist within the Department of Homeland Security Science and Technology Directorate and has conducted research on transboundary animal disease viruses at the Plum Island Animal Disease Center for over 13 years. She holds a graduate degree in Homeland Security with a specialization in agricultural biosecurity and food defense from Penn State University. As a subject matter expert for the design of applied biosecurity studies to assess viral inactivation, much of her recent work has focused on decontamination and disinfection of select agent transboundary animal disease viral inactivation, including African swine fever virus. I think that is one of the important viruses, and the food and mouth disease by chemical and physical means. Prior to her work with DHS, she worked for the US Centers for Disease Control and Prevention, CDC, studying the pathogenesis of arthropod-borne zoonotic viruses. Dr. Gabbard.
Everyone in person and being able to network with everybody. So thank you very much. As stated today, I'm going to talk to you about testing we've been recently completed at Plum Island, looking at the efficacy of ionized hydrogen peroxide for disinfection of laboratory surfaces at the room scale. So to give you a little bit of background on Plum Island, it's a laboratory that is run by Department of Homeland Security, USDA Agricultural Research Service, and the Animal Plant Health Inspection Service. It's in the Long Island Sound off the coast of New York and Connecticut. It's currently the only operational laboratory in the country that can study tier 1 livestock diseases, select agents such as foot and mouth disease virus and African swine fever. Our laboratories are composed of BSL-2, BSL-3, animal biosafety level 3, and BSL-3 enhanced ag vivarium spaces. As many people know, next summer Plum Island will be celebrating its 70th anniversary and is slated to eventually close and transfer its science mission to the new National Bioagrodefense Facility in Manhattan, Kansas. As you can see from my very descriptive timeline here on the bottom, we do not know exactly when this is going to happen. As I said, we're currently the only lab with registration to work with foot and mouth disease virus, so we need to maintain that capability until registrations are in place at the new facility, and there's a lot of politics involved in things like that, so we plan to keep doing the science until they tell us we can't anymore. As part of this transition, we will be tasked with decontaminating certain laboratory spaces at the island, and that is the background where this project kind of begins. So obviously, we have a variety of decontamination challenges based on the age of the facility. There's a mix of office, operations, laboratory spaces that obviously have various levels of risk associated with the pathogens or sort of work that's done there. The photo in the top right there gives a little overview. The areas in red are the levels of highest risk that we're going to have to prove that we've decontaminated as part of the closure of the island and transition of the property back to stakeholders, whether that be New York State, GSA, or the public. Our currently approved decontamination practices that we use day to day, primarily liquid chemical decon with Virkon S, bleach, and sodium hydroxide for pipes. For fumigation, we use paraformaldehyde. That's currently the only fumigant that is allowed to be used on the island and approved by New York State. So one of our key programmatic goals right now is to validate new decontamination technologies to optimize, our ability to both effectively and efficiently decontaminate the laboratory spaces for eventual closure. So this is kind of similar to what I just said, but basically we want to have validated data sets with the pathogens that we use on the island against these technologies. So not using surrogates, to use the actual viruses that are used in the animal rooms primarily. Right now, we don't have a lot of supporting efficacy data to support even the formaldehyde use in the lab. So our leadership has identified some technologies, one of them being ionized hydrogen peroxide as a potentially safer, cleaner, faster, more effective way to decontaminate and potentially replace formaldehyde, which obviously has safety, material compatibility concerns. Just a little bit of background on IHP. It's not necessarily the same as vaporous hydrogen peroxide. I had printed out a apparently old schedule, and I thought the talk right before me was on VHP, so I don't have to compete anymore with that talk, so I was happy about that. I did not individually choose this technology. It was chosen by our leadership, but basically there's various reasons why it makes it more attractive for our specific spaces. It uses a 7.8% hydrogen peroxide concentration versus the 30+% that other companies and products use. It's purported to have better material compatibility. There's no need to precondition the space to meet a certain temperature and humidity requirement, which is important for us because that is a challenge. There's also a lower contact time. So I stole this from the company website. So the company that manufactures this solution is Tomy Steramist, and the actual disinfectant is called binary ionization technology. So that'll be referred to as BIT solution. It's a solution of 7.8% hydrogen peroxide. It's EPA registered. Basically how the technology works is it passes through a sprayer out of a plasma arc, cold plasma arc, which creates reactive oxygen species that are purported to inactivate different microorganisms when they contact them. And then you can see at the bottom there that the other viruses that have EPA registrations for that product. Just a little background on our test organisms. So we have African swine fever and foot and mouth disease virus. These viruses are probably for us the highest risk and that are most used on a daily basis in actual animal challenge studies in our animal rooms. It's actually kind of a nice opportunity because they're very different. So for African swine fever, we have a large enveloped double-stranded DNA virus. The specific strain we're using in our testing is BA71V, which is a virocell adapted strain that we can grow to high titers and concentrate. And then we have foot and mouth disease virus, which is a small non-enveloped coronavirus. It's an RNA virus. We're using FMD strain A24, which is a highly virulent wild type virus. ASF, as you can see from the species listed there, it's going to infect domestic pigs, warthogs, wild Suday, so feral pigs, things like that. FMD, anything with cloven hooves, so cattle, bison, deer, pigs, goats, sheep. Here's a little background on the equipment that's that we purchased from Tomy Steramist. And just as a disclaimer there, they are not sponsoring our study in any way. This is all of the government purchasing the equipment, all the test chemicals and everything for these tests. So the Tomy environment system, which you can see there in the top left, is kind of a big suitcase-like unit that's where you program your settings for your testing. It's kind of easily movable around the room. That's connected by the 3 cords there to the spray ports, which It's kind of covered up there in the top right, but basically it's a sprayer that's on a tripod that can be moved around the room. We purchased motorized heads so that it sprays back and forth during the tests. Each unit is remote controlled, which is very nice for us since we have to shower out of these rooms. We can shower out and then start the machines through the window and watch everything kind of start up. So it creates a visible fog that moves like a gas through the area being treated, which is nice because you can actually see it. Depending on your room size, you can get a complete treatment in 75 minutes. The contact time on the product label is 15 minutes, so 15-minute dwell time. The equipment is kind of set it and forget it. So you program it based on the volume of your room, and then it'll inject 0.5 mils per cubic foot. So if you have a larger room, it's going to take longer to inject that amount. And basically, after the injection, you count down the dwell time and begin aeration. Okay. So this is a picture of our testing room. It's one of our large animal rooms in the BSL-3 ag space. It's about 13,000 cubic feet. We have 2 of the environment systems set up in this room. So there's 6 individual sprayers and 2 machines. It's pretty much rectangular, but there are 2 small rooms, one to house feed and one that has access to the shower. So during the testing, we tape those rooms off so that the volume for the chemical that we're calculating is just based on that room size and does not include any of the HVAC system or anything else. There's 3 fans that are set up in the room, and that's not to disperse the chemical during the injection or dwell time. It's actually to more quickly aerate the room after the test is complete so we can go in and get our samples. So all of our samples are prepared in a BSC in the BSL-3 lab. It's very similar to other quantitative carrier tests that have been presented earlier today. We're following pretty much ASTM 2197 or the OECD test method. We have stainless steel coupons, 1 centimeter, and we also have carbonated concrete coupons that we are going to inoculate with the virus, 10 microliters per coupon. The virus is prepared to contain a 3-part soil load, which is BSA, yeast extract, and bovine mucin. Each of these sample boxes that you can see in the hood there contains both a stainless steel coupon and a concrete coupon for FMD and ASF. We also place a biological indicator in there, which is 6 logs GST stainless steel ribbon sold by Mesa Labs. And we have a chemical indicator strip for hydrogen peroxide that's still sold by Sterad. So those basically all get prepared inside the biosafety cabinet, put into a carrying case, transferred down to our test room, and then applied to our sampling sites throughout the room. For these tests, we had 36 sampling sites around our room, pretty much about 5 feet in between each one, evenly dispersed throughout the room. We tried to, pick high, medium, low parts on the walls. The sample boxes are magnetic, so we can invert them and put them upside down on the ceiling, on the side of things, on the floor. We tried to make attempts to find difficult places to put these for the chemical to reach, but there's not a lot of hiding places in this room. There's no penetrations on the walls. Anyways, I think the next slide these are a couple of our more difficult to reach sampling sites. So underneath the table, on the side of one of the lights on the ceiling, underneath the TOMI machine, inside a water trough, and then just a sample box placed upside down on the ground. So the 4 tests that I will be going over today, all of them have the same dose of the chemical injected into the room. All of them have the same aeration time. There is slight variability in the concentration of the test chemical. And then our main variable that we're changing throughout is just the dwell time. for the chemical in the room prior to aeration. And then if you see here, the bit solution concentration for test number 1, based on the certificate of analysis we received from the company, the concentration was 7.6%. After that, we wanted to test the product at what's called the lower certified limit, which is the lowest concentration the company is allowed to sell their product at. So we had a specially blended batch made that was 7.4% hydrogen peroxide. And then those pictures there kind of give a nice indication of how the fog disperses throughout our test room over time. So after the tests are run, we collect the sample boxes, transfer them back to the lab. All the chemical indicator strips are visually inspected for a color change. The BIs are incubated for 7 days for a positive or negative kill, also based on color change. The virus coupons are placed into 5 mils of tissue culture media, and they are vortexed. We plate 4.5 mils of that initial sample directly onto cells without any cytotoxicity. That is then incubated for FMD for 2 days and for ASF for 7 days. We calculate TCID 50s, and then additionally, after that, any negative samples we are freeze thawing and we passage them 2 more times in tissue culture. And that is a kind of gold standard diagnostic method for determining your sample is completely negative. When you do that, you can release low levels of infectious virus that may not cause typical CPE in your samples, and you will see the results of those passages in the results section. So just quick summary, tests 1 and 2, we're using a 15-minute dwell time, both viruses, both surface types, and we're looking at that 7.6% and 7.4% hydrogen peroxide bit solution. For both tests, every BI was negative at all locations, and we also put them inside of the TOMI machines. That is how our biosafety department deemed that the room could be clear and the equipment removed after these tests. All the chemical indicator strips were positive, so there was good dispersion of the chemical around the test room. You can see in the picture there, we had control boxes in the room that actually were sealed with parafilm, so the test chemical could not reach them. So those strips are positive. The magenta color, the orange color is the color change. Here is an example of virus recovery from our control coupons. So for the blue bars are the wet titer of 10 microliters of each of the test viruses directly into media. For FMD, we're getting 5.4 logs per sample. ASF, we get 6.4. As you can see, FMD is very stable when dried, and this trend continued across all of our tests. ASF, which we already knew prior to this, and which is why we use a higher starting concentration, does not like to be dried on surfaces. So you lose at least 2 to 3 logs just through the drying process, which is a challenge for that virus. But we are still getting 4 logs, which is nice. Just kind of to expand upon that and show you the variability. This is these graphs include additional controls that we brought into the rooms that were also sealed in the parafilm boxes. The take-home message, FMD, still very stable, reproducible across all of them. ASF, a little more variability. All right, so these are the results from the first 2 tests. And I may use the terms complete kill or inactivation. And basically when I say that, it's taking the inverse of those control titers. So 5.4 logs of FMD or 4.3 logs of ASF, and that would be your log reduction. So after 3 blind passages, we consider those samples completely negative without really a lower limit of quantification. So on stainless steel, at a 15-minute dwell time for both concentrations, we see a complete kill for ASF and FMD. So non-porous surfaces were good. We're seeing a complete kill of ASF on concrete. for both chemical concentrations. So from the tables here, you can see our issue was we were getting positives with FMD and concrete. If you look at test number 1 there for passage 1, we actually only had 1 well positive from 36 total samples in passage 1. And as we continue to go by passage 3, we had 8 samples that came up positive. When we look at the 7.4% test 2, we start with 5 positive samples and that goes up to 21. So basically, for our purposes, even though if you were to look at the titer information, these results would correlate with 3 and 4 log reductions, for our purposes and to make stakeholders feel comfortable that we've killed everything in our facility. We really want to see a complete kill after 3 passages. So then it's basically what I just said. We move on to test 3 and 4, where we're looking at the extended dwell times. So we wanted to still use that 7.4% lower certified limit for the product concentration and just see if extending the contact time would result in higher kill. So our dwell times we tested were 60 minutes and overnight, which for us is 16 hours. There's not a lot of intermediate tests we can do based on our workday since we have to take a ferry from work. Even a 1-hour test, we're working a 12-hour day to get these samples analyzed and put in the freezer. 16 hours is really similar to what we use for our formaldehyde protocols we currently have in place. Kind of set it up right before you go home and then come back in the morning and collect your BIs, or whatever you're doing. So here's the results of those 2 tests. You could see with the 1-hour dwell time, we're still getting 6 out of 36 positive after pass 3. When we go overnight, we are seeing a complete kill, no live virus remains. So the conclusions are fairly apparent of that testing. The nice thing about this experiment is the results follow all conventional scientific principles. The agent that should be harder to kill on the surface that should be harder to kill it on is what we saw. And by increasing the contact time, we are seeing a higher inactivation rate. This is a little busy, but it's basically to show that there was not really any correlation between a certain sampling location and its instance of being positive over those 4 tests. So location 1 was not positive every time. It might have been positive in 1 test and not the other. And it wasn't like every sample on the ceiling was positive or anything like that. So it was kind of random. And this is obviously just for the concrete. Those are the only positives that we saw. So in conclusion, we're pretty happy with this data so far. It looks like a promising technology, especially for inactivation of our particular viruses of interest on non-porous surfaces. Since we work with select agents, all surfaces in the laboratory are sealed. There is no unpainted concrete. Everything is painted or sealed or what have you. So that is beneficial for us. And then also the passaging of the virus did increase the sample positivity. So it was useful for detecting very low levels of virus in certain samples. So our next steps, we want to write this efficacy data up into a report and possibly submit it to EPA in support of obtaining registration or a label claim, at least for FMD, and potentially test other chemicals to see if we can get a quicker, more effective kill on the unsealed porous concrete. And thank you to everybody in our lab at Plum Island. It's a lot of, anyone that processes virus samples knows how long that it takes and how many weeks just to get those results. So thank you. We'll take any questions.
Thanks, Lindsay. Great presentation. Excellent work. The one question I have is, it looks like you used a fairly square room with low surface area to volume ratio. I'm wondering if you can speculate on what would happen if you had a more complicated room, like a hallway or somewhere with more surface area to volume, higher ratio?
Yeah, so we picked this room specifically for its simplicity, since what we wanted to demonstrate here was just the efficacy of the chemical getting to those spaces. In initial tests that we did like a year before this, we actually put BIs up in the ductwork and things like that. We did not tape off the rooms. We put them in the shower room, in the feed room, and we got 100% kill with our BIs for that. I think it might be a little bit different if you had a room that had cabinets in it or drawers or something more challenging. But for us, since we're just looking at these technologies as a terminal decon method, most complicated structures and equipment will be removed from our spaces prior to fogging or fumigation or whatever it ends up being.
Yeah, the other point is that peroxide usually does have a tough time with concrete. You may just want to consider spraying with your Vercon or a peracetic acid based product in tandem with the.
Yeah, I mean, we've done liquid efficacy tests on these concrete coupons and we get kill. So they're obviously not a standardized coupon, they're homemade. So there's going to be variability in that surface for each one. There's going to be variability in deposition of our virus inoculum. So
Any other questions?
Good talk. I had a question. Can you recover infectious virus after 16 hours from concrete?
yes, we can.
Okay.
So I didn't go into it in depthly here, but these concrete coupons are carbonated, which lowers the pH of them, which allows survival of virus for longer periods of time to simulate aged concrete in a building. We've demonstrated it. We have environmental chambers that we can look over time, survival of virus. And I think for ASF, which is the pickier of the two, we've shown survival up to 28 days on concrete.
Okay, thank you.
Hours, that's a big gap.
It's a big gap, but it would require people to stay overnight. And for us, since we would never set up an actual decontamination where people have to stay overnight, it's not relevant to us, but it has been brought up. It would be nice to get a more, linear curve. Does it take 2 hours or 3 hours? But it is challenging for us. So
Sure.
what?
I'll camp overnight.
Okay. And it's not just that, we need operations guys up in the maintenance to close the dampers to the room and be available and all. Okay, all right, you're welcome.